Okay, good, it's recording. So today we are covering chapters 8 and 9, starting with chapter 8, obtaining energy. Now, when we're talking about obtaining energy, also feel free to interrupt me and ask questions whenever you need to, because I won't know what y'all don't know. So, starting with chapter 8, obtaining energy. When we talk about obtaining energy, for these next few chapters, we're going to talk about cellular respiration and photosynthesis. Today, I'm only covering cellular respiration because that's all we've covered in class. And when we talk about obtaining energy, the main thing we're going to consider as energy is ATP. Anybody remember what that stands for?
What ADP stands for?
ATP.
Something triphosphate.
Okay, adenine triphosphate.
Yeah.
Adenine triphosphate. Now, if you don't remember what adenine is one of your bases for DNA. So when you draw out an ATP, you should have a 5-sided ribose sugar, a nitrogen base here that is the adenine type, and then you should have 1, 2, 3 phosphorus bases, right? Triphosphate, 3 phosphoruses. Now, when you break the triphosphate there and turn it into a diphosphate, aka 2, it releases a lot of energy. That energy is going to be used for whatever biological reaction you need it to, right? And today, because we are only covering cellular respiration, I'm going to skip the chloroplast part and just cover good old mitochondria. Mitochondria. What do mitochondria do?
Produce energy.
Via, what's the process?
Cellular respiration.
Good. Now, before I continue, okay, hang on. Let me make this red. You got you got this wavy inside. All right, let's label this so that you'll remember. So what is this inside wavy bit called?
Is that the matrix?
No, the matrix is inside of that.
Oh, I feel like I know it, but I just don't.
There's 2 acceptable answers here.
It looks like RNA or something.
It is the inner membrane. That's one of the answers. What's the other name for it? Starts with a C. Nobody knows? It's called a cristae.
Oh my god.
Should have taken that. You should have written that down last chapter when I did review. So that inner folded membrane is called cristae, and that's actually where a significant amount of the cellular respiration happens. Don't forget that your mitochondria can split in half via binary fission to multiply themselves. And you generally find more mitochondria in your nerves and muscle cells. Now, before we continue, let's go over some extra words. Catabolic pathway. Right? When we think of catabolic pathways, right? We try thinking in exergonic versus endergonic. Does anybody remember what exergonic and endergonic mean?
Exergonic is when things exit the cell. Endergonic is when things enter the cell.
That's exocytosis and endocytosis.
Oh my.
Not.
Okay.
I mean, I can understand where you got confused. That's okay. Exergonic and endergonic have to do with the release of energy. Exergonic, exergonic, someone, anybody?
No? Energy.
Yes, it releases energy. I'm going to put ATP in place of energy from here on out. But so yeah, exergonic, you release energy. Actually, you know what? To be more accurate, I'm just gonna put energy. It's probably a better idea. Take back what I said. Exergonic reactions release energy, right? Endergonic reactions do the opposite. You invest energy. Does that make sense? Now, if we're thinking about catabolic pathways in terms of exergonic and endergonic reactions, let's just define one more word before we can help with catabolic pathways. Catabolism. What is catabolism? Anybody? Crickets, man. I got crickets in my class. All right, catabolism is all about breaking big molecules into little ones. We will talk about this more later, but when you break your big molecules into smaller molecules, you generally release energy, right? So catabolic pathways are exergonic. Does that make sense?
Yes.
Okay, good. I'm going to erase all of this bit now that y'all know endergonic versus exergonic. We're going to cover more of endergonic versus exergonic in a little bit, but right now, let's just cover the opposite of the catabolic pathway, anabolic pathways. If a catabolic pathway is exergonic, an anabolic pathway is
Endergonic.
good, the exact opposite. Plain and simple. Cool. Let's talk about exergonic versus endergonic more, and then we will get into enzymes. I'm gonna cut this page in half. Cool. Let's talk about, let's do exergonic in blue. So an exergonic reaction is all about releasing energy, as we have said before. When you draw a graph, if any of you are in chem or were in chem, you would see a graph like this. You would see a flat line at your reactants, and you would see a curve like that for your products. Now this is an exergonic reaction. You start with a lot of energy and you end with a little, meaning somewhere in the middle, energy had to be released, right? When we talk about, what color do I, magenta, let's do magenta. When we talk about endergonic reactions, we talk about investing energy, right? And when you invest energy, same kind of table, you will see this, you will see little energy in your reactants go up, and you will end with a lot of product energy, right? You will always see that curve, it'll go slightly higher than your products and then curve back down. And does anybody remember, which means, sorry, let me slow down a bit, which means at some point energy needed to be invested in order for you to gain energy in your products. Does that make sense?
Yes, thank you.
Okay, now, stop for a second. I'm gonna erase all that. When you look at these graphs, you see the curve? Right. Come on, hang on. There we go, okay. So you see how it curves like this? Right? There is a bit here where it needs to go up before it can release all that energy. Whereas in endergonic reactions, there's still that little bit here where it needs to go way higher than your product, than the energy in your products, but then it curves back down. This section is called your activation energy. Erase the line in the middle. This is activation energy. It is the energy you need to invest in order to kick your reaction into starting. Now, when we look at these normal reactions, usually it'll be a ton of energy you need to invest, right? Whenever you're processing glucose into ATP, you actually require a lot of energy in order for that reaction to start breaking down glucose into its carbons and hydrogens and ATP. Or at least you would if you didn't have a catalyst. So let's make a new graph. When we talk about catalysts, right? Say for instance, I have an exergonic reaction. I start with tons of reactant energy and I fall like that, right? But if I have a catalyst, right, in reality, what it's going to look like, here, I'll do it in red, it's like this. Notice how my activation energy required has decreased, right? Rather than requiring this much energy, I now only require this much energy. I'm not quantifying anything because you really don't need numbers to understand this part, but do y'all get the point, right? If I have a catalyst, it reduces my activation energy. I'll put that word on the side. Reduce.
Marcus,
Yes.
you know how they have that like symbol with like delta and then some letter for activation energy?
Yeah, it's delta H.
Okay.
Yeah, delta H represents your activation energy. If you see that on a question, that's what it represents. They kind of just assume you know that because you took chem, but I'm guessing none of you remember that, so that's what it means. Delta H is that. All right, so now, what is the most common catalyst we're going to talk about? Because we're in biology, we can't use inorganic catalysts.
Protein?
Close. It's enzymes. Let's talk about enzymes.
Bro.
So, enzymes have very specific traits to them. If I can untangle my legs. Here we go. So, when we are talking about enzymes, there are 5 things you need to remember about enzymes. Number 1, they are proteins. All enzymes are proteins, all right? Can't have an enzyme that isn't a protein. Number 2, they are catalysts, meaning they will speed up your reactions. Number 3, what do catalysts do? They
Lower activation energy.
Good. They lower your activation energy. I'm going to represent that with delta H. Oh, someone just joined? Ah, nice. All right, what's that? So, number 3, they lower your delta H or your activation energy. Number 4, they are specific. Your body designs your enzymes to do one reaction and only that reaction. Number 5, anybody remember what the last thing is? Y'all should have this written down. Okay, they're reusable. Right? So once an enzyme does a reaction, it doesn't just die. it'll just keep doing that same reaction because they are reusable, right? Now, let's do more things about enzymes. So, everybody got this part? Yes, no?
Yes.
Okay, good. Now, let's talk about more about enzymes. So, usually when you are shown an enzyme, you'll have like, I don't know, I'm going to represent my enzyme with weird Pac-Man symbols. All right, they don't actually look like that. So this is my enzyme. And in order for an enzyme to perform a reaction, it needs to make contact with, wait a minute, that was really bad, hang on. A substrate. If you're writing this down, the substrate is, hang on, let me erase that bit, that looks weird. The substrate is basically what the enzyme is grabbing, your reactants, to make the reaction, right? So if I have an enzyme that is performing, I don't know, it's performing cellular respiration, right? And it needs to combine, what is it? What are the two things that you need to combine? It's trying to combine, glucose and oxygen, right? The two substrates in this case would be glucose and oxygen. So it'll grab a glucose, it'll grab an oxygen, it'll combine them, and then boom, reaction, right? That's very basic, but everybody understand that? Here, I will just put it.
Yes. Yeah.
Wait, Marcus, can you repeat the meaning of a substrate one more time?
So your substrate is essentially your reactant, right? The enzyme, when it is properly built, it is only designed to do one specific reaction. It'll look for the reactants or the substrates for that reaction and combine them or take them into this specific part, which I will do next. Does that make sense?
Yes, thank you.
All right, cool. So the enzyme sees the substrate, but the substrate, because enzymes are very specific, the substrate must make contact in this part. Notice how they are the same shape. That is because it is very specific, and it must make contact at the active site. Let me draw an arrow. Whoops, that's not sorry. SITE. Right? It needs to make contact at the active site. And when your substrate makes contact with the active site, and the active site will perfectly fit the substrate, when it makes contact at the active site, the enzyme will do its reaction and it will spit out a product. And we don't have a special name for the product, it's just a product. Does that make sense?
So does the enzyme alter the substrate?
Yes, it has to for the reaction. Because you know how reactions work, right? You'll throw in a reactant and you'll get something completely new as a product, right? So no matter what, if the enzyme is doing a reaction, the substrate will be altered. Sometimes you'll also have enzymes which just break your substrate down, and that's still a reaction, but they will usually be altered. I say usually because sometimes you have malfunctioning enzymes, but then your substrate doesn't connect.
Marcus, is there a name for this process?
Is there a name for this process? No, this is just how enzymes work. Now, let's talk about what affects enzymes, because enzymes are proteins, and proteins are weird. So, factors which affect your enzymes. Number one. Wait a minute, I shouldn't be on the highlight function. Hang on. Number one is enzyme concentration. Oh dear, oh dear. So, enzyme concentration. Why do you think enzyme concentration affects the way your enzymes work? Anybody want to whip out their critical thinking skills?
Like the more or less of an enzyme might depend on how much will break down or something.
Kind of. So here, I will put my y-axis as the reaction rate, and my x-axis as the concentration of enzymes. And when I put a graph like this, it's gonna do that. That should be a straight line at the end. I'm sorry that it curved. So if the graph looks like this, and we see this, I shouldn't do it in yellow, my bad. Should do it in red. When we see this, as it begins to plateau, we consider this enzyme to be saturated. So at this point, come on. So at this point, the enzyme is saturated, right? Why do you think we would say that? And why is the reaction rate plateauing? Nobody? Okay, let me use a different example, right? Say, for instance, you're doing a group project. Now, this group project requires 2 people, but you have a group of 6. Are 4 people actually doing the work?
No, the 2 smartest kids are probably carrying.
Exactly, right? In this case, there is too much enzyme that not all of them are actually doing any work, right? So now as your enzyme concentration increases, there's only so much substrate you can use to continue having that reaction, right? More enzymes is good, but at a certain point, you will literally just run out of stuff for them to do, right? This also applies if you're not looking at enzymes, if you're looking at the substrate concentration, right? If I'm looking at, come on, there we go. If I'm looking at the substrate concentration, right, at some point it'll plateau because there's only so much substrate I can add and that the enzymes can keep up with, right? Because remember, the body only produces so many enzymes. And if there's too much substrate, the enzymes only work at a certain rate. So no matter what type of reaction, whether or not you're looking at the enzymes or the substrates, one of the things will be saturated at a higher concentration. Does that make sense? Can I get a yes or no?
Yes.
Thank you. Is everybody not confused?
Not confused.
Okay, so no need to go over anything again? Okay, good. Cool, let's keep going. That is factor number 1. Number 2, let's talk about temperature. Now, if we remember our proteins work the best at specific temperatures, right? So when I look at a graph and I see a curve like this, and here we're at like, I don't know, 37 degrees, right? And this is the reaction rate. What do you think this graph tells me about this enzyme?
And
It likes body temperature.
Yes, it likes working at 37 degrees Celsius because the reaction rate is at its highest at 37 degrees Celsius. If the temperature goes too low, your enzymes will slow down. So like this side. If it goes too high, what do you think happens?
They will still slow down.
It
Becomes
begins to denature.
denatured?
Yes, it begins
Oh.
to denature. It doesn't just slow down, it literally starts breaking apart. And the thing you need to remember is the tertiary structure, or the 3 and then the degree symbol, is what gets denatured. Anybody remember what the tertiary structure is? It's when your alpha helix or your beta sheet begins to form bonds with itself and begins folding. So if your tertiary structure begins to denature, it starts to unfold back into its secondary structure. All right, everybody good?
Yes.
Okay, good. So number 3, pH. Another graph, because who doesn't love a graph? Same thing on the y-axis. By the way, I should be copying these graphs down. They really make the notes really easy. And at the bottom, it's the pH, right? And I'm going to put 2 different enzymes on here. We'll do one in magenta, because a curve looking like this. And then let's do one in blue. This curve looking like this. I'll say this one's at 7, this one's at 2. So if we're looking at pH, what's the difference between the magenta enzyme and the blue enzyme?
One likes working in more acidic environments.
Which one?
The magenta one.
Good. The magenta one loves working at acidic environments. Where do you think I would find this one?
In the stomach.
Yes, because stomach acid, right? So each enzyme is unique, right? Certain enzymes will enjoy being in more basic, certain enzymes will enjoy being in neutral, and some enzymes will enjoy being in acidic areas, right? But if they get out of their ideal pH range, they will stop working and they will begin to denature, right? And in this case, for pH, their secondary and their tertiary structures will begin to denature.
Do you know if we have to memorize which structures break down?
I would.
Okay.
Ooh. All right, everybody understand this part? pH is pretty easy to understand.
So Marcus, if their pH, like in the magenta one, if it's
Mhm.
low, its secondary and tertiary structure will denature?
If it gets out of its ideal pH range, then yes. Like, for instance, if it becomes too basic, or even if it becomes neutral, it'll just start to denature.
What about the quaternary structure and stuff?
The quaternary structure? Okay, if your secondary or your tertiary structure denatures, your quaternary is already gone. So usually we don't account for that, just because if your tertiary is gone, your quaternary is already gone.
So as long as it stays somewhere between, like let's say 1 to 3 for the magenta, it's fine. But if it's out of that, it denatures.
Yes. All right, everybody ready to move on to number 4?
Yes.
All right, let's do number 4. Number 4. What does the word salinity mean?
Salt concentration.
Yep, it's all about salt. This one follows a similar graph. You got your concentration of salt here. Standard stuff, there's a hill. And it functions best at some arbitrary number. And y'all get the idea, right? It functions at a specific, it functions the best at a specific salt concentration. Not too difficult to understand, right? For this one, if your salinity gets too high or too low, your secondary and tertiary structures will begin to denature. Standard stuff, standard stuff. Do I need to stick around on this one for too long? Or
No,
one-
it sounds about
yeah,
the same as the
this
other
one
one.
is just like pH, right? Just with a different thing. So now let's talk about number 5. 5 and 6 kind of go hand in hand. 5 is all about activators. What do activators do?
Do they start the reaction?
They not quite. Enzymes start the reaction. Activators.
I don't know, Chief.
Start something. They start enzymes. Say, for instance, the enzyme is like a machine. Each machine has an on button. That's what the activator is. Right? It will activate the enzymes to start doing their things. Now, there's 2 types of activators. You have cofactors. And you have coenzymes. Cofactors are inorganic. Coenzymes are organic. Does that make sense?
Can you list some examples of like
Cofactors.
and enzymes, coenzymes?
Coenzymes. What's a? Oh, we have one. Hang on. Let me flip through my notes real quick. Yeah. Coenzymes. NAD and FAD are coenzymes.
What are those, Marcus?
We will get into those in a second, but you'll understand them later. So, y'all got that though? Cofactors, pretty simple. They're like vitamins. Vitamins help out your enzymes. Coenzymes are like other organic compounds that help your enzymes do their shit. It's my recording. I get to cuss. Woohoo! All right, ready? Everybody good?
Yes.
All right, let's keep going. Number 6. If we have activators, we also need, if I have an on button, what else do I need?
Off button.
Okay, inhibitors are kind of like an off button. So your inhibitors reduce enzyme activity. Hang on. Now, when your inhibitors, they decide to reduce that enzyme activity, right? There's 4 different kinds, right? And your body will do some of them. Others will be poison. And I can't really name any examples right now, but certain drugs can be inhibitors for your enzymes to stop doing reactions.
Excuse me, Marcus.
Yeah.
You're kind of going in and out.
Oh, am I?
Yeah.
All right. Is this better?
Yes.
Okay, sorry. I was leaning away from my mic. So, if we're looking at inhibitors, the first kind is competitive. Your competitive inhibitor will affect, hang on. Okay, y'all remember what that is, right? That's how I represent an enzyme. Enzymes have this spot. What is that called?
Activation site.
Good, it is the active site. A competitive inhibitor affects, I don't know, let me blue, your active site, right? What your competitive inhibitor does is it jumps into the active site, which stops the substrate from fitting properly. Notice how that competitive inhibitor is in there. Now that pizza slice will never fit into Pac-Man's mouth, and poor Pac-Man will never be able to eat the pizza. Does that make sense?
Yes.
Yeah. The important thing to note is that the competitive hits the active site. So here, let me label. So if it hits the active site, right, there is a way to deal with competitive inhibitors. And that is very simple. Add more substrate. Why does it work? I don't know. It just works.
Wouldn't the substrate not fit on the site?
Your competitive inhibitors won't affect every single enzyme. Remember, you have multiple. So if it's blocking one, just add more. Let the other ones pick up the pace. Does that make sense? Can I move on to the next one?
Yes.
Okay, cool. So now that we've covered competitive, we talk about, hang on, let me erase all that. The next kind of inhibitor is non-competitive. Or allosteric. And you'll understand why they're called that in a second, but let's redraw Pac-Man. Good old enzyme Pac-Man. You'll understand why I put that gap there in a second. Your non-competitive inhibitor is different from your competitive inhibitor because it hits this spot. That's not the active site. That is the allosteric site. So when it, dang it, let me rewrite that. So when it hits the allosteric site, what it'll actually do is change the shape of the enzyme itself. So let me redraw good old Pac-Man. Poor Pac-Man now only has, oh, wait a minute, this is in blue. I should have done it in black. Give me 1 second. Poor Pac-Man barely has his mouth open. And our old substrate is now too wide to fit into the enzyme, meaning no reaction is going to happen, right? Since it hit the allosteric site, it changed the shape of the enzyme, so now the enzyme cannot receive the substrate. Does that make sense?
Yes.
And I don't have a solution for this one written down, so you might want to Google that. But yeah, so in this case, the enzyme is now completely useless, unless it removes itself from the allosteric site. But if it's a foreign thing that isn't supposed to be in your body, it'll just bind there and just stick forever. Now, let's do the third type. We have competitive, we have non-competitive. Now we have, oh wait, I need to erase non-competitive and allosteric. Now we have irreversible. What does irreversible mean?
Can't be changed.
Yes, it is permanent.
So there's no solution to this one, huh?
Yeah, they kind of just hit your enzyme and your enzyme's useless. Now, for irreversible, they can either be competitors. Oops. Hang on. Or allosteric. So I'm not going to draw a picture for this one. What's the competitor? It binds to the...
Active site.
Yep. Your allosteric binds to the...
Allosteric site.
There you go. Does that make sense?
Yes.
Yes.
All right. And last but not least, I'm going to have to clear the whole screen. So make sure you got this written down, memorized, if you have photographic memory, which isn't real. All right. Last thing we need to talk about is feedback inhibition. Feedback inhibition. So when your body tries to convert something into something else via a reaction, right? Sometimes it'll need multiple enzymes to do the job, right? So say for instance, I have compound A and I'm trying to get it to become compound E. So A needs to, so what the body will do is it'll have an enzyme here to turn A into B. Then I'll have another enzyme. We'll call them 1, 2, to turn B into C. Then I'll have another enzyme to turn C into D, and then another enzyme to turn D into E. Notice that to turn compound A into compound E, I need 4 enzymes in this chain to just convert A into E, right?
Is this electron passport or transport? I mean,
Can be, but this is a completely different example, right? It's not just electron transport. Your body does this for multiple things. Like say, for instance, you're lactose intolerant, right? If you're lactose intolerant, there's something wrong with the enzyme chain that makes your body not be able to break down lactate. Say, for instance, enzyme 3 is missing from that chain when your body tries to break down lactates. So then your body can't break down them cheeses and milks very easily because it's missing this entire half of the chain, right? And it makes you poop, right? So when your body decides that it has made enough of compound E, it wants to stop the chain. The easiest way for it to stop the chain is to just turn off enzyme 1. Just kill it, right? Just turn it off. Now the whole chain doesn't work, and now you're not producing compound E. Because your body needs to regulate how much of everything it makes, it can just turn off enzyme 1, boom, the whole chain is just not working anymore, and you don't have an excess amount of E. Does that make sense?
yes.
Anybody confused?
No.
All right. I think the lactose intolerance example is probably the best way to explain that. All right. Now we are finally done with enzymes. Are there any questions on chapter 8? No. Let's talk about cellular respiration. Cellular respiration is exergonic. All living organisms must perform cellular respiration. That includes plants. And it is a form of hydrolysis, meaning you are going to start breaking things down, And before we explain anything else, let's go over redox. What is redox?
Gaining electrons?
What is redox okay, what is a redox reaction? It's about what and what? Oxidation and reduction. Right? If you oxidize, what happens?
You lose electrons.
Good, you lose an electron. Oxidation is loss. If you reduce, what happens?
Gain.
Right, reduction is gain. Or my favorite acronym, oil rig. Oxidation is loss, reduction is gain. So let's use an example. So when we're talking about good old, what is it? C6H12O6 plus 6O2 becoming 6CO2 plus 6H2O plus ATP, right? What is getting oxidized and what is getting reduced? Can anybody figure it out?
Is glucose getting oxidized?
Not all of glucose entirely. The carbon, oh wait, I've made that too far. The carbon is getting oxidized. Now, why is it getting oxidized? Well, when we are looking at oxidation and reduction in biology, the easiest way is to follow the H. In this case, the H is going to behave like an electron, right? If you lose electrons, you are being oxidized. Notice how on the left side, the carbon is connected to the hydrogen, but now on the right side, it is no longer connected to the hydrogen. So if the carbon is being oxidized, what is being reduced?
Hydrogen.
Not quite.
Oxygen?
Yes, oxygen is being reduced. Notice how on the left side, the oxygen is not bound to any hydrogen. But on the right side, it is. Now I'm guessing some of you are confused. Remember, hydrogen in this case is going to behave like an electron. Just pay attention to where the hydrogens are. Notice how on this side, hydrogen is with the carbon, and on this side, it isn't. Notice how on this side, the oxygen has a hydrogen, or the oxygen is not connected to a hydrogen, but on this side, it is. Does that make sense to everybody? Hydrogen itself is never going to be oxidized or reduced. It's whatever it is connected to. Make sense?
Yes.
Yes, okay, good. Now that everybody understands that, let us define one more word. Actually, you know what? We can define that word later because it is on the last step. But everybody understands the redox, correct?
Yes.
Good. The next thing we need to talk about is electron carriers. Electron carriers, we've brought them up before. They are NAD and FAD. NAD and FAD are coenzymes which pick up electrons during the process of cellular respiration. when they pick them up, they will be reduced. NAD and FAD will always be reduced into NADH and FADH2. Now, you're going to ask, why is this important? We will understand that later. But does everybody understand this part?
Wait, Marcus, you said that NAD and FAD pick up what?
An electron. They are
Okay.
electron carriers. They pick up an electron. And you're probably confused because hydrogen has a positive charge and it shouldn't be an electron. Remember, in this case, hydrogen will behave like an electron. Does that make sense?
Yeah, that makes sense.
Okay, cool. So, when your body does cellular respiration, it will actually release some electrons in the form of H ions, right? H plus ions. And those H ions, now electrons as we're going to call them, will connect to NAD and FAD for a future purpose, right? That future purpose will be at the very end of the cellular respiration cycle. So just stay on the lookout for these things. But now that we are, I just clear all this. So now let us go over how to perform cellular respiration. To perform cellular respiration, there are 3 steps. Number 1, glycolysis. Number 2, the Krebs cycle, or as it is now called, the citric acid cycle. And number 3, the electron transport chain. The electron transport chain is the final step. Everybody got this written down?
Yes.
Okay, cool. So let's talk about glycolysis.
Is glycolysis the first step?
Yes, glycolysis is the first step. The first step in doing cellular respiration is glycolysis. Glycolysis is all about breaking the glucose in the cytoplasm of the cell. This does not happen in the mitochondria. Write that down. This does not happen in the mitochondria. And roll your R when you do it. Now, when glycolysis happens, you need to invest 2 ATP. So in order to perform glycolysis, you actually need to spend some energy, but what you get out of it is 4 ATP, meaning your net total is only 2 ATP. Right? Does that make sense?
Yes.
All right. So now, let's talk about how glycolysis actually works. When glycolysis happens, you are going to receive a 6-carbon molecule. Do you know what it is?
Is it NADH?
No, it's glucose. 6 carbons.
Bruh.
It's glucose. First, you have glucose. Now, why do we perform glycolysis? Well, glucose is actually too big to fit into the mitochondria. So first, I need to break it into 2 separate molecules, right? These molecules are called pyruvate. And when I break my glucose into 2 molecules, this is where I'm going to spend my 2 ATP. And I'm only going to follow the path of one of my pyruvates, but I will just, actually, you know what, I'll draw the other one too, right? So when your pyruvates split in half, let me recenter this glucose molecule. It's bugging me. Hang on. Give me 1 second. When I break that glucose molecule in half, I'm going to get pyruvate, a 3-carbon molecule with a little phosphorus on the end. Same thing down here with a little phosphorus. Now, oh, hang on, let me spell out pyruvate for you guys. That is pyruvate. Pyruvate is then going to release one of its electrons. And when it releases one of its electrons, to phosphorylize, meaning it's going to gain another phosphorus, your NADH or your NAD is actually going to pick up that extra electron and become NADH. Same thing happens on this side. That NAD is going to become NADH because it phosphorylized, right? When you phosphorylize, you gain phosphorus. And when it phosphorylizes, what it's going to look like, it's going to look like this. So you got your 3 carbons and you got a little phosphorus on the end. Same thing on this side. There's a phosphorus on one end, 3 carbons in the middle. There's a phosphorus. Whoops. There's a phosphorus on that end. Does that make sense?
Yes.
Okay, cool. So, hang on, wait, let me review my things real quick. All right, so when these phosphorylize, they will actually be able to convert 2 ADP into 2 ATP. So we spent 2 at the beginning. We've made 2 here. We've made 2 here. Now we have 4 ATP. Oh, whoops, might be. So now y'all should understand where I got my extra 2 ATP from. Does that make sense? And now I have 2 molecules of pyruvate and 4 molecules of ATP at the end. So I have net gained 2 molecules of NADH. Hang on, wait, let me do this in a different color to show you all the products. Red. I now have 2 molecules of NADH at the end, 2 molecules of pyruvate, and 4 molecules of ATP. But since I spent 2 at the beginning, I only gained 2. So those are the products of glycolysis. Does that make sense? Geez, I'm going to have a hard time drawing the electron transport chain.
Can you repeat that one more time?
Okay, so when I perform glycolysis, I am going to spend 2 ATP to make 2 molecules of NADH, 2 molecules of pyruvate, and 4 molecules of ATP.
Can you explain why it like splits into 2?
Because I spent 2 ATP to cut it in half.
Okay.
To show you the power of flex tape. Okay. Anybody else still confused?
Wait, Marcus.
Yes.
The proton thing, I'm not sure what I heard, but I heard something about an electron, and then I heard something about a proton. Can you just like explain that again?
Okay, which part? The phosphorylation step?
Yes.
Okay, so when you first split your pyruvate, or when you first split your glucose into pyruvate, they only have 1 phosphorus. You want 2 phosphoruses on your pyruvate. So to do that, you phosphorylize it, and when you phosphorylize it, pyruvate actually gives off 1 electron, right? When you give off that electron, the NAD will show up and pick up that electron and become NADH. Hence, this part.
Where does the extra phosphate come from?
Your cell, the cell will just have it floating around. It'll pick it up because the cell membrane needs to absorb some phosphorus.
Okay.
Does that make sense? And when that electron flies off, you will also have enough energy to turn 2 ADP into 2 ATP.
And this happens in fermentation too?
We will go over that at the end.
Okay.
Fermentation is a bit confusing, so. All right, everybody ready to move on to step 2?
Yes.
All right, step 2. So now, when we hit step 2, we are entering the mitochondria. And let me once again draw this mitochondria. And this time, it's going to happen inside the mitochondrial matrix, meaning here's your inner membrane. This is where the Krebs cycle happens. And then you have another membrane on the outside. All right. So the Krebs cycle actually happens inside of the mitochondria. So when we talk about the Krebs cycle, oops, sorry. For these citric acid cycles. The first thing we need to do is take our old molecule of pyruvate. I'm only going to represent it with the 3 carbons. I'm going to ignore the phosphoruses I added earlier. And I need to turn it into something called acetyl-CoA, or a 2-carbon molecule. Now I can't just magically turn a 3-carbon molecule into a 2-carbon molecule without doing something weird. So what happens is a CoA thingy, I don't actually have a picture for it, will enter right at the end, right? And during this process, you're actually giving off a CO2 and an electron. And when I give off an electron, my good old friend NAD plus will come by, pick it up, and become NADH. Does everybody understand this first step? This is the very first step.
Yes.
All right, so to turn pyruvate into acetyl-CoA, it'll release an electron, it'll release CO2, and it will accept a CoA thingy. I don't exactly know what that is, and to become a 2-carbon molecule called acetyl-CoA, which will then help me do this process. And to be, wait, no, I'll go over products in a second after I explain the process. So, ready for me to move on?
Yes.
All right. So when your acetyl-CoA goes through the Krebs cycle, it'll actually, so here, let me draw my boy acetyl-CoA. And it goes through the cycle. Notice it's a loop. Acetyl-CoA will go through this loop until it runs out of electrons to give, right? And acetyl-CoA, first, as it moves through the cycle, it'll give off CO2, or 2 CO2s. When it does this, it'll also release 3 electrons. Which means, what happens when I release electrons?
NAD plus.
There you
I was
go.
supposed to-
NAD plus shows up. Oh, whoops. And it becomes NADH. And there's 3 of them, so I make 3, right? 3 NADs, 3 H's, 3 NA. I put NA. NADH. There we go. And as it's doing this, ADP, one ADP molecule will show up. And as it's going through the cycle, it'll pick up a bit of energy and become ATP. All right, good luck, Alex. I will send the recording out to anybody who wants it. So ADP will show up as it's going through the spinny, and it'll become ATP, right? And along with this, as it's going through the cycle, it's going to give off 2 more H's. Now, I've talked a lot about NADH, but this time, since there's 2 of them, FADH will show up. FAD plus will show up and pick up these 2 H's to become FADH2. And then it'll loop and loop and loop until it runs out. And by loop and loop and loop until it runs out, I mean it goes twice.
So Marcus, so there won't be 2 FAD2 plus, it's just 1?
Yeah, it'll only be 1 because FAD can actually pick up 2 electrons. Right, so it'll actually do this loop twice, and then we'll pick up another acetyl-CoA and it'll do it again, right? And so what's the point of doing all this if I've only gained 1 ATP? Well, in reality, since it does it twice, everybody, hold on, wait, has everybody drawn this model?
Yes.
Yes, okay. So if you have this model or screenshotted it for reference, I'm gonna clear this in, I don't know, 10 seconds. Five, 4, 3, 2, 1, and it's gone. All right, now let's talk about products of the Krebs cycle. Right, so here's your products. So if you're looking at that model, what I made 2 CO2. I made 1 ATP. I made 1 FADH. And I made, what is that, 3 NADH? And I do this cycle 2 times. Multiply that up. I get 4 CO2, 2 ATP, 2 FADH2, and 6 NADH. Those are my products. So right now, I have produced 2 ATP from glycolysis and 2 ATP from the Krebs cycle. That's not a lot of energy. So now we get into the electron transport chain.
Wait, Marcus, I thought it produces 4 ATP from glycolysis.
Yeah, but you have to spend 2 to start
Oh,
it.
you spend 2. Okay, and then
Yeah.
your total, the net totals. Okay.
Yeah, so your net total is only 2. So right now I only have 4 ATP produced, right? Not a lot of energy. Can't function the human brain off of that. So I need to do the electron transport chain. Now, if you're looking at the original reaction for cellular respiration, I have glucose, I add oxygen, I produce carbon dioxide, and I produce water. I don't know where water has come from yet. Water is going to come from the electron transport chain. So let's talk about the electron transport chain. And I'm about to cry because I'm about to draw something that looks ugly as hell. All right, hang on. Give me like 5 minutes. Oh, no, wait, I already screwed it up. Okay. Do that. All right. Does anybody know what this is supposed to represent? Anybody?
The
Any
cell
clues?
membrane?
Close. This is the inner membrane of the mitochondria, right? There's an outer membrane somewhere, but this is the inner matrix, or this is the inner membrane, and inside of this is the mitochondrial matrix. My weird curvy handwriting there. But yeah, this space is the mitochondrial matrix. This is the space between the inner membrane and the outer membrane, and this little bit right here I've added is the outer membrane. Does that make sense? Do y'all know where we are now?
Yes.
Yes.
All right, good. Now that we know where we are, let's talk about what it does. So this part is called the electron transport chain. Write that down. And when you do the electron transport chain, this is all about making water and ATP. So the first thing that shows up at the very start is NADH. NADH, what it's going to do is it's going to go up to this protein. It's going to exit, but it's going to exit as an NAD+. And I'm going to represent the traveling of the electron with an orange arrow. The electron that NAD gives off will go here into this protein. And hang on, wait. And also at this protein, let me switch back to black. FADH2 is also going to show up. It's going to hop in, it's going to hop out as in FADH, or sorry, no, not FADH, FAD2+. Right? And it's also going to give off its electrons into here. These electrons will flow up through this thing, through here, and they are going to travel down and accept their place with, and they will meet, okay, hang on. H2 plus 1/2 O2. And they will meet here and produce water. Okay, to summarize what I have drawn, because I didn't explain it very well, NAD shows up and it decides to give off its electron into the chain at this protein. FADH also shows up and it'll give off its protein at this, or give off its electron at that protein, right? So that part makes sense, right?
Yes.
Okay, continue following the orange line. Those electrons will continue moving through the protein chain until they hit the fourth complex. That's what I'm going to call it. The fourth complex. The fourth complex is where it'll funnel those electrons to meet H2 and an oxygen. Oxygen, write this down. Oxygen is the final acceptor for electrons. Let me write this down for you, with you guys as well. Oxygen is the final electron acceptor. What does this mean? It means when that oxygen meets that electron, it'll form H2O, and now that electron has made its way into water. And this is where we produce water from cellular respiration.
Marcus, so would you say that NADH and FADH2 are the first and second electron acceptors?
Yes, they are the ones who accept the electron and transport it around. And oxygen is the last one to receive it because that's where the water is, right? Now, why does this matter? Because when oxygen does its thing and it picks up that electron, what it'll actually end up doing is, to preface, there is a bunch of, let me do this in dark blue, H plus ions floating around here. And when that electron meets H2 and oxygen, it'll flow out that way. And the reason it does that is because when that ion passes through that protein, it is actually combining the electron, the hydrogens, and the oxidants together to make H2O. Does that make sense?
But it makes sense, but like, why are there electrons floating around?
Look, I honestly, I don't know because she didn't cover that part, but just know that they're there.
Thank you.
And so what happens is when it shoots out, right, a bunch of them are going to shoot out because this doesn't just happen at one place, it happens everywhere on the cell membrane. When that happens, you will actually form a high concentration of H plus molecules in this space, right? So there's going to be a bunch of random H plus ions just floating out here. Now, why is that important? Because the fact there are this many just floating out here means they can perform the last step. You take, because right now we've only made water. We haven't made any ATP, right? You take one phosphorus and you combine it with an ADP. What happens when I combine phosphorus and literally anything?
Energy.
Big word I just taught you guys.
Phosphorylation.
Yes, it goes through phosphorylation to form ATP. But you gotta get that energy from somewhere, right? Well, if you have a high concentration gradient on the outside and a low concentration gradient on the inside, it goes through diffusion, right? Your H plus, any of them floating on the outside, would just go through here. And when they come down, they will they will phosphorylize ADP and that extra phosphorus into ATP. And that is where you produce ATP from when you do the electron transport chain. Does that make sense?
So there's a diffusion of electrons or the H plus coming in to the last one.
Yes, they're following their concentration gradient. They are going from a high concentration on the outside to a low concentration on the inside to make ATP. And then, since there's still more electrons flowing around, they'll get shot out so that they can produce H2O. And that is how you make ATP with this. Now, how much ATP do you make? You make roughly 34. there is enough energy like going through all of this to make 34 ATP.
So in total of after everything we have 38.
Hang on, wait, 6. It should be between 34 to 38, yeah. The generally accepted number is 36, but none of the numbers for the electron transport chain are concrete, so it can range pretty far. But just know it's around 36 to 38. If they ask you on a test, just put 36. So in total, 36 is made. And I will summarize all that up at the end. But this whole process of H plus flowing in due to the concentration gradient is called chemiosmosis. Chemiosmosis is all about that concentration gradient allowing us to make ATP, right? And I have a specific definition around here somewhere. I wrote it down like 5 minutes ago. Hang on. Where did I write it down? Not here. Chemiosmosis. It is the movement of ions down their concentration gradient, meaning from high to low. So with that, does everybody understand this whole process?
Yes.
All right, cool. Last thing I need to tell you all about, it's the fifth complex, the final molecule, this thing. Maybe I should do it in a different color. I'll do it in red. This thing, that thing is called, let me erase chemiosmosis now that you all know what that means. This thing is called ATP synthase. Why is it called ATP synthase? Because it synthesizes ATP. It is a protein that synthesizes ATP. All right. That makes sense?
Yeah.
Yes.
All right, last thing to remember before I erase this entire picture. Here, everything is being oxidized. So if there's a test question and it asks you what happens to NADH and FADH at the start of the electron transport chain, they get oxidized. All right. So, ready? Everyone's ready for me to clear?
Yes.
All right.
Oh, never mind.
So let's do one final summary of the products. At the end, glycolysis makes 2 ATP because you use 2 to make 4. 4 minus 2, you make 2. Krebs makes about 2 ATP. And your electron transport chain makes about 32-ish ATP, meaning you have a total of 36 ATP per 1 glucose. And that is a lot of energy. Does that make sense?
Yes.
All right. Since I have made all that energy, let's go over anaerobic respiration. That's the last thing we need to go over for today. And that's pretty simple. Anaerobic respiration is what happens when you have no oxygen, because you can only do this half due to oxygen. Your anaerobic organisms stop at glycolysis. Glycolysis is the only thing they can do. They can just snap a glucose in half, make some energy, make 2 ATPs worth of energy, and then call it a day, right? So when we talk about anaerobic respiration, respiration without oxygen, right? You can't do the Krebs cycle and you can't do the electron transport chain. So what can you do? You can do something called fermentation. When you do fermentation, there's 2 kinds. What's the most common kind? Y'all should know it.
Alcohol?
Yes, alcohol fermentation. Because the first thing a civilization figures out once they've mastered farming, they make alcohol. Fun fact, if y'all didn't know that. So, in this case, when you break a glucose into pyruvate, the anaerobic organism has nothing to do that pyruvate with. So it's got to figure out something to do with it, right? And in this case, pyruvate is converted into ethanol. Oops. And this releases some CO2. So it's going to release some CO2. And ethanol, if you ever look at ethanol, it looks like this. Here, let me do it in blue. Or is that a double one? No, CH3. That is what ethanol looks like. So if you ever see that, that's an ethanol. And you know that it has done alcohol fermentation. Oh, man, my voice is going to give out soon. All right, everybody understand alcohol fermentation?
Yes, sir.
Yes.
All right. So now we go over the other kind of fermentation, lactic acid fermentation.
Marcus?
Yes.
So for alcohol fermentation is when the pyruvate can't really do anything, right?
When you do fermentation, the pyruvic acid or the pyruvate can't do anything. So in alcohol and in lactic acid fermentation, it's all about converting pyruvate into something else. Does that make sense?
This is due to the lack of oxygen?
Yes, thank you.
Yes, this is due to the lack of oxygen. So now we have lactic acid fermentation. And you guys experience lactic acid fermentation when you're running around and your muscles start to burn. And it's just like, oh God, my hip, I'm dying. And that's because you're not breathing properly and not getting oxygen to the cells that need it. So when the cells don't have oxygen, they start performing lactic acid fermentation. They start turning your pyruvate into NADH. Like straight up, they just turn your pyruvate into NADH. And this causes the formation of lactate, which happens to exit your cell, land on your muscles, and start causing them to burn. So, y'all ready to have another picture?
Yes, please.
All right, now that we have that definition down, let's do another picture. Picture time. Let's do glucose in green today. So I have my glucose. Glucose is broken down into 2-pyruvate. Y'all remember the process of how glucose is broken down into pyruvate? When it does that, 2 ADP gets to phosphorylase into 2 ATP. And when glucose makes pyruvate, right? Your main thing now is that pyruvate gets to produce 2 NADH. So that 2 NADH will give off an electron to produce 2 NAD+. Your pyruvate will take advantage of that electron's production and make lactate, which looks like this. Oh dear, hang on, wait. Do it in red. Hang on, this is a long molecule, so. And then that electron from your NAD plus, or sorry, your NAD pluses go back into that chain and cycle back and forth, back and forth, back and forth, making a bunch of waste that gets ejected. Does that make sense?
Yes.
All right, everybody's good?
And
So it can't,
is this all within glycolysis?
yes, because you can't do the Krebs cycle or the electron transport chain if you don't have oxygen.
Thank you.
And that is the end of chapter 9. Any questions?
No.
All right, that's it, we're done. Now is the part where I try to figure out whether or not it actually recorded. I should have just had one of y'all record. Makes it easier on myself. All right, let me stop sharing. That is it from me.
Marcus, may you please share?
All right, any questions?
I'm good.
Everybody's good? All 5 of you? No questions, really? Okay, well, if everybody's good, I will see y'all tomorrow, either in school or online. Hopefully y'all are understanding this better than on Thursday when you learned it. Yeah, I think that's it. I will probably be back to do photosynthesis, which we're starting tomorrow, I think. And that's it.
All right, thanks, man.
All right,
Thank you,
see
Marcus.
y'all.